So what is the advantage of immobilization? So immobilization helps protection of enzyme from degradation and deactivation. It helps retention of enzyme and enzyme free products, recycling or repetitive use of enzyme, its cost efficiency, enhanced stability of the enzyme, used as a controlled release agent, and its ability to stop the reaction rapidly by removing the enzyme from reaction, and it also have advantage in the development of multi-enzyme system. So the immobilization process have a number of advantages, and because of these advantages it is important to study the enzyme immobilization methods and their significances. So what is the property of an ideal carrier matrix for enzyme immobilization? The first property should be that must be inert. The matrix should be physically strong and stable. The matrix should be cost effective. Matrix should be regenerable, so you can regenerate it, you can reuse it. It must be biocompatible. It must be easy to make derivative of it, which will help in the immobilization of enzyme to the matrix. Its mean parameter diameter and swelling behavior, and it must enhance the specificity of enzyme. If it not enhance, it may not decrease the specificity of the enzyme, but if it acts as an enhancer, it is more better, and it reduce the product inhibition. So in this table we have different natural polymer, synthetic polymers and inorganic materials that are used as a carrier matrix. Natural polymers consist of alginate, chitosan, chitin, collagen, gelatin, cellulose, starch, pectin and saccharose. The synthetic polymers includes ion exchange resin, diacellulose, polyvinyl chloride, polyethylene glycol, nylon, etc. The inorganic materials includes silica, zeolites, glasses, charcoal, aluminium, etc. So in this way we have a number of natural, synthetic and inorganic materials or polymers that are used as a carrier for the matrix which are used in immobilization. Let us discuss smart polymers. So what is smart polymers? They are novel stimulus responsive carriers. So they undergoes dramatic conformation change in response to a small change in their environment like temperature, pH and ionic strength. For example, poly N isopropyl acrylamide, 2-methyl, 2-methoxy, ethoxy, ethyl, methacrylate, methacrylate, and oligo ethyl glycol methacrylate. So these are the different examples of a smart polymer. So significance of a smart polymer is not only there in enzyme immobilization, that smart polymers also have significances when you are talking about the drug delivery. For example, a particular drug is encapsulated in a smart polymer that must be delivered to a specific tissues or a specific cells. So when we are taking that smart polymer that is encapsulating the drug, so because of the different physiological conditions of different tissues, they are going to change its conformation and delivering drug to a specific tissue. So smart polymer is not only important for enzyme immobilization, but they have n number of significances. So let us now discuss what is the properties of enzymes, carriers and reaction system for efficient immobilization. The size of enzyme, that is important. The conformational flexibility required for the mechanism is important. Its surface properties is also important. Surface properties such as charge density, presence of hydrophobic and hydrophilic region, glycosylation and its isoelectric point. These properties helps you to design a particular matrix or design a protocol which help you to tag it with the carrier or with the matrix. So the carrier which are used in enzyme immobilization includes organic or inorganic carriers, hydrophobic or hydrophilic carriers. It must be a stable, it have surface area, porosity, and its particle size is very important. When you are talking about the carrier, the carrier must be compatible with enzyme as well as with matrix. So they must have different properties based on the requirement or based on the characteristic of your enzyme. And we will also have different reacting system, such as the medium where the reaction is, where the immobilization is operating is important. Its diffusion limitation is also important. Its enzyme inhibition, that means where we are performing reaction, is that particular condition is having some enzyme inhibition properties or not, that is very important, because when we are immobilize a particular enzyme in a matrix, that condition or reacting system does not have any inhibitor present for that enzyme. So viscosity and any other non-acidic solute support interaction, they all are very important. So when we are using enzyme for immobilization, enzyme properties, carrier properties and reacting system properties, they all are having very significant role in the process of enzyme immobilization. What are the different immobilization methods that are used? So immobilization method is based on following principles. It may be based on adsorption, it may be based on covalent bonding, it may be based on ionic bonding, it may be based on cross-linking, it may be based on entrapment, it may be based on affinity binding, it may be based on metal-linked immobilization. So different types of enzyme requires different immobilization methods. At the same time, based on our specific requirement, we may change the immobilization methods. The first immobilization method is adsorption method. This method is simplest, oldest and reversible method. So adsorption of enzyme and the carrier can proceed by different type of interaction, such as hydrogen bonding, hydrophobic interaction and van der Waals forces, and its common support material is activated charcoal, alumina, ion exchange resin, cellulose, lignin, etc. So what is advantage of adsorption method? The first is it is easy to perform and cheap. The second is enzyme can be immobilized into the native state, because we are here only adsorption property of enzyme and matrix are utilized, and so no activation is required. It is wide applicable and capable for high enzyme loading. But the disadvantage of this method is desorption of enzyme resulting from change in the pH, temperature and ionic strength, and absorption because it is a not induced absorption, so that is very slow process if it is taking place spontaneously. So the second method is covalent binding. So the covalent binding of enzyme to this matrix is a classical method, and it involves direct attachment of enzyme to the support matrix through covalent linkage. It provides a strong and stable attachment as they are formed through reaction between functional group of supporting matrix and enzyme surface that contain amino acid residues. It can be used on unmodified protein since they rely only on the naturally present functional group. Use of spacer arm may provide greater degree of mobility to the enzyme to show its higher activity, and its common support matrix which are used here is polyacrylamide, porous glass, agarose and porous silica. So what is derivatization or activation that is required for covalent bonding? So in this figure you can see how a cyanogen bromide method and cyanogen activated saffrose is used for the activation of your enzyme or immobilization of your enzyme to a matrix. You can see then how cyanogen bromide is activating saccharose and how your enzyme is tagged to your specific matrix. There are some other methods that are also used for covalent binding, such as ethyl chloroformate, which is less toxic to the cyanogen bromide. You can see how ethyl chloroformate is utilized in enzyme immobilization. There is other molecule, that is carbodiamide, that is also used for enzyme immobilization by using the covalent binding method. There are other molecules like glutaraldehyde, which is also used for enzyme immobilization. Glutaraldehyde is well known fixative, so it cross-link between amino groups and carboxylic group. So you can see that how this glutaraldehyde is going to be involved in covalent bond formation between enzyme and the matrix. So what is the advantage of this covalent binding? So this covalent binding is a tight binding of enzyme to support matrix, and enzyme leaching in aqueous medium is minimized because they are covalently attached to the matrix. So when you are using it, then there are less chance to wash out, and it is a simple and mild method with a number of applicability. But what is the disadvantage of this method? Because enzyme is covalently modified, this covalent modification may alter its enzyme activity. It may causes denaturation of the enzyme during covalent binding, and only a small amount of enzyme can be immobilized. If you have excessive enzyme, then they cannot be immobilized. The next method is entrapment. So in entrapment method, this is one of the best immobilized strategy to avoid any negative influence on the enzyme structure. The enzyme are immobilized by encapsulation or entrapment in synthetic or natural polymeric networks. Encapsulation of enzyme prevents their denaturation, and entrapment can be activated through gel or filter entrapment or micro encapsulation. Solid gels or solution gels widely used, and highly porous silica materials extensively used for protein immobilization, particularly for development of biosensors. So entrapment can be further classified into lattice type and microcapsule type. In lattice type, entrapment involving entrapping enzyme within the interstitial spaces of cross-linked water-insoluble polymers. In microcapsule type, entrapping involves enclosure of enzyme within semi-permeable polymer membranes. You can see here how an enzyme is entrapped in a matrix and how it is going to be entrapped in the droplet. When you are entrapping into the matrix, it is very important when you design a biosensor, and entrapping into the droplet is going to have different significances. So what is the significance of entrapments? So entrapment is fast, cheap, and mild conditions are required for reaction process. So therefore enzyme native state is maintained. It's a large surface area between polymer and enzyme, and loss of activity upon the immobilization is minimized. But what is the disadvantage of it? It has limitation in mass transfer, and there may be some time chances of enzyme inactivation after the encapsulation, because once it is encapsulated, then we have more chance to undergo enzyme inactivation. In other words, you can say that during the process of encapsulation, there may be the loss of enzyme activity. The next method is cross-linking. So irreversible. So what is cross-linking? Cross-linking is irreversible carrier-free enzyme immobilization. So enzyme acts as its own carrier. So, the formation of intermolecular cross linkages between enzyme molecule by means of bi or multifunctional reagents such as glutaraldehyde, nanodimetric supports such as electrospun nanofibers improve its process efficiency. So, these material are increasing the cross linking. These material are increasing the process of cross linking. So, what is cross linked enzyme aggregate? This is prepared by first aggregating the enzyme in precipitants such as acetone, ammonium sulfate and ethanol, followed by addition of cross linkers such as glutaraldehyde. So, what is advantage of cross linking? So, cross linking have very little enzyme desorption because they are covalently attached. It is best used in conjugation with other method, but disadvantage is the cross linking may cause significant changes in enzyme active sites. The next method is metal linked immobilization. So, transition metal salts and its hydroxide are also participated in immobilization. So, deposited matrices can not occupy all coordination sites of the metal. Therefore, some sites are free. So, metal such as titanium and zirconium salts, with the help of supporting matrix like cellulose, chitin, silica based carriers, they are used in the immobilization. So, we will have immobilized metal ion affinity absorbent, that is also known as IMA absorbents. So, we have chelators ligand via EDPA can immobilize first onto the solid support by means of stable covalent bond, and then metal ion are then bound by coordination. So, metal linked immobilization is simple. An immobilized enzyme have a specific activity which is relatively high, and you can achieve a 30 to 80% specific activity when we are using metal linked immobilization. But the disadvantage of this method is its operational stabilities achieved are highly variable and results are not easily reproducible. What are the factors that influences the performance of immobilized enzyme? So, now we have learned what are the different techniques that are involved in enzyme immobilization. Let us discuss what are the factors involved in performance of enzyme immobilization. The first factor is hydrophobic partition. And what is effect of this on the enzyme immobilization? Enhancement of the reaction rate of hydrophobic substrate. The second is microenvironment of carrier, and its effect is hydrophobic nature stabilizes enzymes. So, multi-point attachment of the carrier, it enhances enzyme thermal stability. Then spacer arm, it prevents enzyme deactivation by providing the mobility to the enzyme. Then diffusion constraint, this basically affects enzyme activity decrease and stabilizes of the enzyme. And then presence of substrate or inhibitor, it increases higher activity retention. Then physical post treatment, it improves enzyme performance. And different binding modes, it's alter the activity and stability. The physical structure of the carrier, its activity retention is also dependent on pore size and physical nature of the carrier. So, this carrier with large pore size migrate diffusion limitation and leading to the higher activity retention. So, in this way we can see that how the different factors are involved in the process of enzyme immobilization and how they are influencing the process of enzyme immobilization. Understanding the different immobilization technique and the factor influencing immobilization, let us have discuss the significances or application of immobilized enzyme in industrial processes. The first significance is biomedical applications. The second is food industry application. The third is biodiesel production. The fourth is bioremediation. And this immobilized enzyme also have significances in textile industry, and they also have significances in detergent industry. What is the limitation of enzyme immobilization? The major limitation of enzyme immobilization is the cost of carrier and immobilization, change in the property or selectivity of enzyme that is used in the immobilization, because once the selectivity or activity or enzyme efficiency of enzyme is altered, then it will basically alter the product formation. So that is one of the major limitation of this enzyme immobilization. The third one is mass transfer limitation. The fourth one is problem with cofactor and its regeneration, because in the biological system we can easily regenerate that cofactor, but when we are talking about immobilized enzyme, it is difficult to regenerate it. The fifth limitation is with the problem of enzyme immobilization with multi-enzyme systems. And the last one is during enzyme immobilization there is a loss of enzyme activity.